Nephron is the basic structural and functional unit of the kidney. Its chief function is to regulate the concentration of water and soluble substances like sodium salts by filtering the blood, reabsorbing what is needed and excreting the rest as urine. A nephron eliminates wastes from the body, regulates blood volume and blood pressure, controls levels of electrolytes and metabolites, and regulates blood pH. Its functions are vital to life and are regulated by the endocrine system by hormones such as antidiuretic hormone, aldosterone, and parathyroid hormone. In humans, a normal kidney contains 800,000 to 1.5 million nephrons. Structure: two general classes of nephrons are cortical nephrons and juxtamedullary nephrons, both of which are classified according to the length of their associated loop of Henle and location of their renal corpuscle. All nephrons have their renal corpuscles in the cortex. Cortical nephrons have their loop of Henle in the renal medulla near its junction with the renal cortex, while the loop of Henle of juxtamedullary nephrons is located deep in the renal medulla. They are called juxtamedullary because their renal corpuscle is located near the medulla. The nomenclature for cortical nephrons varies, with some sources distinguishing between superficial cortical nephrons and midcortical nephrons, depending on where their corpuscle is located within the cortex. The majority of nephrons are cortical. Cortical nephrons have a shorter loop of Henle compared to juxtamedullary nephrons. The longer loop of Henle and juxtamedullary nephrons create a hyperosmolar gradient that allows for the creation of concentrated urine. Each nephron is composed of an initial filtering component and a tubule specialized for reabsorption and secretion. The renal corpuscle filters out solutes from the blood, delivering water and small solutes to the renal tubule for modification. Renal corpuscle, composed of a glomerulus and the Bowman's capsule, the renal corpuscle is the beginning of the nephron. It is the nephron's initial filtering component. The glomerulus is a capillary tuft that receives its blood supply from an afferent arteriole of the renal circulation. The glomerular blood pressure provides the driving force for water and solutes to be filtered out of the blood and into the space made by Bowman's capsule. The remainder of the blood passes into the efferent arteriole. The diameter of efferent arteriole is comparatively less than that of afferent arteriole, increasing the hydrostatic pressure in the glomerulus. It then moves into the vasa recta, which are only found in juxtamedullary nephrons and not cortical nephrons. The vasa recta are collecting capillaries intertwined with the loop of Henle through the interstitial space, in which the reabsorbed substances will also enter. This then combines with efferent venules from other nephrons into the renal vein, and rejoins the main bloodstream. The Bowman's capsule, also called the glomerular capsule, surrounds the glomerulus. It is composed of a visceral inner layer formed by specialized cells called podocytes, and a parietal outer layer composed of simple squamous epithelium. Fluids from blood in the glomerulus are filtered through the visceral layer of podocytes, and the resulting glomerular filtrate is further processed along the nephron to form urine. Renal tubule: the renal tubule is the portion of the nephron containing the tubular fluid filtered through the glomerulus. After passing through the renal tubule, the filtrate continues to the collecting duct system, which is not part of the nephron. The components of the renal tubule are: proximal convoluted tubule, loop of Henle, descending limb of loop of Henle, ascending limb of loop of Henle. The ascending limb of loop of Henle is divided into two segments. Lower end of ascending limb is very thin and is lined by simple squamous epithelium. The distal portion of ascending limb is thick and is lined by simple cuboidal epithelium. Thin ascending limb of loop of Henle, thick ascending limb of loop of Henle. Distal convoluted tubule. Function: the nephron carries out nearly all of the kidney's functions. Most of these functions concern the reabsorption and secretion of various solutes such as ions, carbohydrates, and amino acids. Properties of the cells that line the nephron change dramatically along its length. Consequently, each segment of the nephron has highly specialized functions. The proximal tubule as a part of the nephron can be divided into an initial convoluted portion and a following straight portion. Fluid in the filtrate entering the proximal convoluted tubule is reabsorbed into the peritubular capillaries, including approximately 2/3 of the filtered salt and water and all filtered organic solutes. The loop of Henle, also called the nephron loop or the loop of Hundley, is a U-shaped tube that extends from the proximal tubule. It consists of a descending limb and ascending limb. It begins in the cortex, receiving filtrate from the proximal convoluted tubule, extends into the medulla as the descending limb, and then returns to the cortex as the ascending limb to empty into the distal convoluted tubule. The primary role of the loop of Henle is to concentrate the salt in the interstitium, the tissue surrounding the loop. Considerable differences aid in distinguishing the descending and ascending limbs of the loop of Henle. The descending limb is permeable to water and noticeably less impermeable to salt, and thus only indirectly contributes to the concentration of the interstitium. As the filtrate descends deeper into the hypertonic interstitium of the renal medulla, water flows freely out of the descending limb by osmosis until the tonicity of the filtrate and interstitium equilibrate. Longer descending limbs allow more time for water to flow out of the filtrate, so longer limbs make the filtrate more hypertonic than shorter limbs. Unlike the descending limb, the thin ascending limb of loop of Henle is impermeable to water, a critical feature of the countercurrent exchange mechanism employed by the loop. The ascending limb actively pumps sodium out of the filtrate, generating the hypertonic interstitium that drives countercurrent exchange. In passing through the ascending limb, the filtrate grows hypotonic since it has lost much of its sodium content. This hypotonic filtrate is passed to the distal convoluted tubule in the renal cortex. The distal convoluted tubule has a different structure and function to that of the proximal convoluted tubule. Cells lining the tubule have numerous mitochondria to produce enough energy for active transport to take place. Much of the ion transport taking place in the distal convoluted tubule is regulated by the endocrine system. In the presence of parathyroid hormone, the distal convoluted tubule reabsorbs more calcium and secretes more phosphate. When aldosterone is present, more sodium is reabsorbed and more potassium secreted. Atrial natriuretic peptide causes the distal convoluted tubule to secrete more sodium. In addition, the tubule also secretes hydronium and ammonium ions to regulate pH. Collecting duct system. Each distal convoluted tubule delivers its filtrate to a system of collecting ducts, the first segment of which is the connecting tubule. The collecting duct system begins in the renal cortex and extends deep into the medulla. As the urine travels down the collecting duct system, it passes by the medullary interstitium, which has a high sodium concentration as a result of the loop of Henle's countercurrent multiplier system. Though the collecting duct is normally impermeable to water, it becomes permeable in the presence of antidiuretic hormone. ADH affects the function of aquaporins, resulting in the reabsorption of water molecules as it passes through the collecting duct. Aquaporins are membrane proteins that selectively conduct water molecules while preventing the passage of ions and other solutes. As much as 3/4 of the water from urine can be reabsorbed as it leaves the collecting duct by osmosis. Thus the levels of ADH determine whether urine will be concentrated or diluted. An increase in ADH is an indication of dehydration, while water sufficiency results in low ADH, allowing for diluted urine. Lower portions of the collecting organ are also permeable to urea, allowing some of it to enter the medulla of the kidney, thus maintaining its high concentration. Urine leaves the medullary collecting ducts through the renal papillae, emptying into the renal colossus, the renal pelvis, and finally into the urinary bladder via the ureter. Because it has a different origin during the development of the urinary and reproductive organs than the rest of the nephron, the collecting duct is sometimes not considered a part of the nephron. Instead of originating from the metalephrogenic blastema, the collecting duct originates from the ureteric bud. Juxtaglomerular apparatus. The juxtaglomerular apparatus is a specialized region of the nephron responsible for production and secretion of the enzyme renin, involved in the renin-angiotensin system. This knot occurs near the site of contact between the thick ascending limb and the afferent arteriole. It contains 3 components, the macula densa, juxtaglomerular cells, and extraglomerular mesangial cells. Clinical significance. Because of its importance in body fluid regulation, the nephron is a common target of drugs that treat high blood pressure and edema. These drugs, called diuretics, inhibit the ability of the nephron to retain electrolytes, thereby increasing the amount of urine produced. However, this clinical significance raises many concerns about treatments to such medical conditions. Because diuretics are drugs that stimulate the body's metabolism to higher intensities, primarily stimulating the heart, the clinical significance creates minor controversy considering how individuals with high blood pressure would avoid introducing medications that raise their already high blood pressure. Additional images. See also, nephrology, urology, renal tubular acidosis, references.